So at this time, I'd like to call up to the stage Dr. Nambuie and Candace Haddox. Dr. Nambuie is from Stanford University, and Dr. Candace Haddox is from Dana-Farber Cancer Institute, who will provide an update on the state of the GIST research.
All right, thank you, everybody. Thanks for coming to this conference, and really happy to share the update on the state of the art for GIST research. So quickly about us, my name is Nambuie. I'm one of the medical oncologists here at Stanford, 20 minutes down the street. And my research interests are sarcoma clinical trials, genetics, and informatics as applied to big data sets.
And I'm Candace Haddox. I'm from the opposite coast, 6-hour flight away, from Dana-Farber Cancer Institute in Boston. I'm also a sarcoma expert medical oncologist. Also see GIST patients regularly, and my research is in phase 1 trials and biomarker development. So today we'll just go through a little bit of background on GIST. A lot of this is probably review for many of you, but just want to set the stage for the trials that we'll be talking about. We'll go over the INSIGHT trial, the PEAK trial, SARC-44, Strategist 1, which is the IDRX study, and NewBay study, NB003. So, as this group knows well, GISTs are one of the most common sarcoma subtypes, and about 80% of them have mutations in the KIT gene. Well, let's zoom in on that comment for a moment, and I'll break it down a little bit more. So a gene is a segment of DNA. It's made up of smaller chunks of DNA called exons. The exon is basically the zip code within the gene of where something is located. So if you have an exon 11 or an exon 9, that's literally telling you within that sequence where the mutation has occurred. Genes are blueprints for making proteins and molecules that the cells use. So the KIT gene translates down the road into an actual functional thing in the cell that gives its behavior, its function. And in the case of GIST, when there's a KIT mutation, that causes that KIT protein to be stuck in the on position. So if we zoom in on that idea a little bit more, these are some cells shown here at the bottom, and if we take a look at the surface of the cell, we see that the KIT molecule or protein is in the cell membrane. So part of it is in the outer surface of the cell, part of it is inside of the cell. And its job is to receive those green signals and then transmit a message to the cell that says grow, survive, multiply, etc. This is very well controlled in the normal situation. So KIT is a normal functional protein in our cells. But when it's mutated, the message is stuck on. So regardless of how much green signal there is, it's constantly giving that message to grow, survive, and multiply. In the case of GIST, the most common primary mutations are located in exon 9 and exon 11. That's, again, just the part of that KIT molecule where the mutation has occurred. And this makes it susceptible to various drugs that have been approved. The first approved drug for GIST is imatinib, and then these were subsequently approved in the order shown here. Sunitinib is classically second line, regorafenib third line, ripretinib fourth line. But what we know is that on treatment, there can be additional mutations in the KIT gene. So the primary, most often exon 9, exon 11, is the first mutation that arises in GIST. But on treatment, you can get additional mutations that show up in the KIT gene. These can be in the exons 13, 14, or 17, 18. Those are just some of the most common locations that the resistance mutations occur. And those resistance mutations make the KIT molecule change such that the drugs no longer work. So that's why we see resistance to imatinib eventually, because these second mutations occur, and then imatinib no longer can bind and shut down that growth signal. So as we've learned more about GIST over the years, we've found that sunitinib may work best in GIST with exons 13, 14 resistance mutations, while regorafenib and ripretinib seem to work best for exon 17, 18 resistance mutations. This might make one think, well, maybe we should just tailor our treatments to what mutations we find in each individual patient. And that's a research question that's ongoing right now. Currently, we still treat patients in the order that's shown here, imatinib, sunitinib, regorafenib, ripretinib for the most part. And treatment is overall not necessarily tailored to those resistance mutations that come up, but that's what we hope to answer soon. To make things a little more complicated, within a single person, they may have a whole variety of different resistance mutations that come up. So what you're seeing here in this diagram is a stomach and a liver, and the stomach has a primary GIST tumor in it that has exon 11, and the other tumors in the liver have different resistance mutations. Each, 13, 14, 17, 18, they may have a variety. So it's hard to cover all of these mutations with our current treatments. Like I said, 13, 14 is best hit by sunitinib based on research in the lab, while 17, 18 seems to be best targeted by ripretinib and regorafenib. So you might think, well, if there are so many resistance mutations piling up, why not just combine 2 drugs that covers all of them? Well, to date, that's been a challenge because of side effects. If you put ripretinib and sunitinib together, or sunitinib and regorafenib together, you can get a lot of side effects. So that is something that is ongoing in research now, is to find a safe regimen that best targets all of these mutations. We've also learned more about KIT resistance and the mutations that come up over time by checking ctDNA. So what is ctDNA? It's circulating tumor DNA. Literally, you can detect fragments of the tumor's DNA circulating in the bloodstream by doing just a blood test. That's called a liquid biopsy. And this tends to track with the cancer's activity. So we can see high levels of ctDNA in the bloodstream when the tumors are very active, and that can drop off when the tumors are being well controlled. And then in addition to detecting disease activity, we can also get a sense of what mutations are rising and falling over time on treatment. So this has allowed us to understand a lot more about how GIST become resistant, and what we know is that over time, over treatment, we see that ctDNA levels increase for patients on second line, most likely sunitinib, versus patients that are third or fourth line. And then also the number of resistance mutations increases over time. There was a study recently showing that for patients that were at second line or greater, they had over 50% of patients had many resistance mutations, even up to 14 different resistance mutations. And as a research community, we're still understanding how to address that, how to suppress all of these GIST mutations that are coming up. So the ongoing trials now are addressing a lot of these key questions in GIST treatment. Does the specific resistance mutation matter when selecting treatments, or should we just go in the order that the drugs were approved, which the INSIGHT trial is going to inform us about? That one is comparing sunitinib versus ripretinib. Is ctDNA helpful in selecting the right treatment for the right patient? That will also be addressed by the INSIGHT trial. Can we safely combine drugs that more broadly cover all of those resistance mutations? And that will be addressed in the PEAK trial and SARC-44. And then are there single drugs that could overcome all of these resistance mutations? And those are being developed, and Dr. Bowie will go over the Strategist 1 study and the NewBay study. So starting off with the INSIGHT trial, the INSIGHT trial is a global phase 3 study, and it's randomized, so patients receive either ripretinib or sunitinib, and that study is only enrolling patients with exons 17 or 18 resistance mutations. So they have a KIT exon 11 primary and then a detected 17 or 18 resistant mutation. This study is enrolling now. Some of the key eligibility, like I mentioned, is you have to have an exon 11 mutation and only imatinib previously. So patients that have had a lot of different therapies wouldn't be eligible for this study. But for patients that imatinib has stopped working for, they would be able to enroll. There's a pre-screening component where a patient would come in, sign consent to start the research testing process. And in this pre-screening part, they just get a blood test to check that circulating tumor DNA, and they look for the specific KIT mutations. If anything else other than KIT exon 11, 17, or 18 are detected, then they're not eligible for this study because they're only capturing the patients with exons 11, 17, and 18. So if they have that profile, then they're eligible and they can go on to the randomization. That means that there's an algorithm that will decide 1/3 of patients will get sunitinib and 2/3 of patients will get ripretinib, and it's sort of randomized with these, 1/3 versus 2/3, so that there's sort of bias to enrolling more patients into the ripretinib arm. The patients on the study will continue treatment as long as it's helping, and if they're randomized initially to sunitinib and the sunitinib stops working, then they can cross over, meaning they can just start on the other arm receiving the ripretinib treatment. So this study will help us to understand whether the specific resistance mutations matter when selecting treatments, and also if ctDNA is helpful in selecting the right treatment for patients. Next, the PEAK trial. So this is a phase 3 randomized study. It's comparing CGT-9486, also called bezuclastinib, with sunitinib. And it's for patients with GIST, although in later parts of the study it's been restricted more to patients that have only progressed on imatinib, just like the last study as well. And this, I should say, this study is sponsored by Cogent Biosciences, who's here today. So bezuclastinib was sort of tailor-made to target those exon 17, 18 mutations in the KIT gene, and it works really well targeting those GIST cells. And in the earliest trial that was testing bezuclastinib, by the way, at the time it was developed by Plexicon, so that's why it has the name PLX9486 and older data that were released. So it was studied both by itself and also in combination with sunitinib. And what you're looking at here is called a spider plot. Each individual line represents an individual patient, and it's tracking their tumor size over time. So you can see if it starts at 0 and goes down, that means the tumors are getting smaller, whereas if it's upward, then that means the tumors are getting larger despite the treatment. And what you can see is that with the combination, which is the blue lines, a lot of lines are going down or staying stable, and it's a very durable effect, meaning the patients were able to stay on the treatment for a long period of time. And I should mention that this is a population of patients, a lot of them had prior treatments. A lot of them were even third line or greater. So seeing this activity, especially for patients that had already progressed on sunitinib, is really exciting, and that's what led to the enthusiasm of combining bezuclastinib with sunitinib. As I mentioned, it was previously manufactured by Plexicon, and then it moved over to Cogent. It was then named bezuclastinib, and then bezuclastinib was reformulated. If any of you are familiar with the early stages of the study, it took a lot of pills to get up to the recommended dose, so they reformulated it to make it a bit more concentrated. Then they had to do additional testing to make sure that that reformulated version matched up with what we were seeing with the older version. So that was part 1A of the PEAK study. And then they found that the old drug at 1,000 milligrams was equivalent to the new formulation at 600 milligrams. And then part 1B of the PEAK study looked at drug interaction. So they're seeing how do sunitinib and bezuclastinib affect each other. And then we also had the phase 3 part open, which is a randomized part, which I'll get to in a moment. But just looking at these initial parts, part 1A was confirming the dose. So they checked 2 different doses of the bezuclastinib. And ultimately, as I mentioned, bezuclastinib 600 was the recommended dose. And in part 1B, they looked at a series of patients that got bezuclastinib first, then sunitinib, or sunitinib first, then bezuclastinib, to see how that changed how the body metabolized each drug. Overall, with the data from this part, 80% of patients benefited, meaning that their tumors were either significantly smaller or stable. And again, like I mentioned, this is mostly for patients beyond the second line of therapy. Patients that were in their first line of therapy or had only progressed on their first line of therapy, 100% of them benefited. So very exciting data. And of course, subsequently, the PEAK trial phase 3 opened. patients were randomized to get sunitinib or bezuclastinib plus sunitinib, and they can continue treatment as long as it's helping. There's also crossover in this study, so if patients were randomized to sunitinib alone, they can cross over to get the combination therapy. So this trial will answer the question, can we safely combine these two TKIs, or did we address that issue with the side effects? And then also, is it better to have a more comprehensive therapy as the second-line treatment instead of just marching down the usual lineup of the FDA-approved drugs? So these hopefully will be wrapping up the PEAK trial soon and ultimately answering these questions as soon as the data are all analyzed, gathered, and looked at and reviewed. And hopefully we'll get some previews of that in coming research meetings. Next, I want to talk about SARC-44. This is very similar to the PEAK study. This is a trial that I'll be leading, and I want to highlight especially that it's supported by the LifeRaft group. So we have very generous philanthropic funds from Dana-Farber, Cogent, and then the LifeRaft group was also a contributor, which is a really special contribution that helped put on this trial. We just opened to enrollment, and I'll tell you a little bit more about the types of patients that will be enrolling. So we're taking adults and that have GIST with a KIT mutation, so 9, 11, and then for some of the more rare primary mutations, like some patients have a 13 or 17 to start. Those patients can also consider enrollment. Patients must have had prior imatinib and sunitinib, but there's no upper limit or excluding patients that have been on other drugs like IDRX or NewBay or some of the other studies. So patients in SARC-44 will start with bezuclastinib alone for 2 weeks and then add in the sunitinib, and they can continue that treatment as long as it's helping. This will tell us how effective the combination is, not just in second line, which is being studied by the PEAK trial, but later on in therapy after patients progress on sunitinib. And then another key thing that SARC-44 will address is why this combination may or may not work. So we're adding in a lot of additional research and collaborating with lab investigators, and we're collecting ctDNA throughout the study that will help us to see how those mutations rise and fall on the treatment, which mutations seem to be most susceptible, which do we start to see if the treatment doesn't work. And then for a subset of patients, only 20, which will primarily be at Dana-Farber, patients will have a PET scan at baseline before starting treatment, after starting the bezuclastinib, and then after getting the combination. And that will help us to see which tumors are lighting up and becoming dim based on the treatment that they're on. Patients on that group will also have a biopsy, and it's a very directed biopsy so that we get the most information about why the treatments work or don't work. And those biopsy tissues will be sent to one of our lab collaborators that's going to be doing a lot of testing downstream to understand how the KIT activation is changing on treatment and whether that activation predicts resistance. So now I'm going to turn it over to Dr. Bowie to talk about some of the other studies that are ongoing now.
So we've had some really exciting new developments in GIST just presented this year at the major ASCO conference. And I wanted to talk about 2 of them. One drug is by IDRX and one is by NewBay. So this was a first-in-human study of the IDRX-42 drug in metastatic or unresectable GIST. This is a highly selective, potent inhibitor of many KIT exon mutations, including 9, 11, 13, and 17. And if you look at the small chart at the bottom, that basically shows in mouse models how much the tumor is shrinking while on this drug, as in the x-axis is time. And you can see that the IDRX, which is the red and orange and yellow ones, are significantly below the imatinib, as well as the control drug for pretty much all mutations, especially 9, 11, 13, 11, and 11, 17. So this was a phase 1 dose escalation that was presented this year. They treated 73 patients across multiple dose levels, and in the phase 1, we're trying to find the right dose, so we have to escalate it with multiple different patients. As you can see, this is the demographic information, and a lot of patients had been heavily pretreated. Most patients had a median of 4 lines of treatment, which are pretty much all the FDA-approved options. The KIT mutation status was also variable. Some had just exon 11, some had exon 11 and a 13, some had an exon 11 and 17, and some had all 3, as well as having exon 9. And this is what we call a waterfall plot, which is showing how much of the patient's tumors are shrinking. And each bar is a patient. And the numbers that go down are pretty much the percent of tumors that are shrinking. So the further it goes down, the more effective the drug is. And so you really want a steep waterfall. You don't want things going up, you want things going down. And you can see that a lot of patients across many dose levels, which were the different colors, were going down. And if you look at the amount of patients that had a major shrinkage, what we call a major partial response, it was about 23% across all patients. And if you just look at second line, it's about 43%, which is, again, small numbers, only 14 patients in second line, but 43% is really high. If you look at Sutent, which is approved in the second line, it's probably 10% or less which have a response rate. So, this is exciting that we have, more potent KIT inhibitors that are potentially going to land in the new space. They also looked at ctDNA, which Dr. Haddox talked about, and you can see that a lot of the ctDNA numbers for many different exons go down, which show that this drug is very effective across many different KIT exon mutations. Here you can see exon 8, 9, 11, 13, 14, and 17, with almost all of them going down. And here's a radiograph picture of a patient who had exon 9 and exon 17, had 6 lines of therapy, including imatinib, sunitinib, regorafenib, cabozantinib, and regorafenib after rechallenge, showing a significant shrinkage of the tumor. You can see here also that they also checked ctDNA, and they saw that these levels of the ctDNA, which they measured in the blood, went down, which shows that this drug is effective against many different KIT mutations. And this patient is currently on treatment for over 10 months, so it looks like some of these responses are quite durable. We're always wondering about side effects because patients will be on this drug for some amount of time, and so these cumulative side effects can be an issue for quality of life. And you can see that these side effects are a little bit different. So patients do actually get a lot of diarrhea. Sorry, where's my mouse? Here it is. Diarrhea, nausea, decreased appetite, fatigue. Not too much of the hand-foot syndrome that we see in our other TKIs like sunitinib or regorafenib. You do get the periorbital edema and the peripheral edema, but only at about 15% of patients, which is less than imatinib. So this trial is currently open and recruiting right now. This is the IDRX 42 trial. It's now going to open for many different cohorts. So for those first line for treatment naive, second line for progression on imatinib, third line or further with progression on imatinib sunitinib, and third line or further with progression on more experimental therapies such as bezuclastinib, NewBay, or the THE-630. So this represents a really promising drug, which hopefully will find a space in the new GIST armamentarium. And then the second drug I wanted to talk was the NewBay drug, NB003, which is also enrolling now. So this drug is also a single agent, and it's a new, potent, and selective inhibitor of both KIT and PDGFR. Importantly, it does not inhibit VEGF, which is a pathway called vascular endothelial growth factor, and can lead to tumor control, but also can lead to a lot of toxicity that you see with sunitinib and regorafenib, including hand-foot syndrome, hypertension, thrombosis, as well as wound healing impairment. And these are kind of cell line experiments showing across the different inhibitors how much of the target does it hit. And so you want these drugs to hit KIT, and the lower number is the better. So you can see that all these drugs, imatinib, sunitinib, regorafenib, avapritinib, and ripretinib, all hit KIT really well because all these numbers are really low. But when you look at VEGF, which is where you get a lot of those side effects from sunitinib and regorafenib, including the hand-foot syndrome, which really limits it for a lot of patients, you can see that this new drug, which used to be called AZD3229, but it's been acquired and renamed, actually has a pretty high number, which means it has little activity against VEGF as compared to sunitinib, regorafenib, which have very lower numbers, which means it does hit VEGF and gives you those side effects. And here you can see this is basically a chart showing how these different inhibitors respond against resistance mutations. And again, the lower numbers are better, and they're highlighted in green here to kind of give you a better visual cue. And you can see here that the AZD drug, or the NewBay drug, hits a lot of the primary mutations, including exon 9 and 11, but also the resistance mutations in exon 13, which is the ATP binding pocket, as well as exon 17, which is the A loop. And you can see that it's better than certain things like sunitinib, which hits exon 13, but really doesn't hit exon 17, or avapritinib, which hits certain ones in exon 17 and 13, but not as comprehensive. So this is kind of a very good data showing that this drug can be effective across many different types of mutations and resistance mutations. But the question is, how does it work in patients? And so this was presented at the ASCO meeting this year. A small number of patients were enrolled in the phase 1 dose escalations, about 42 patients across many different dose levels, so from 3 milligrams all the way to 40 milligrams. So again, we have to not hang our hat too much on the absolute data because a lot of patients are treated at different dose levels. And you can see here in the demographic information that a lot of these patients were pretty heavily pretreated. A lot of patients had, the most number was 4 number of prior TKIs, which again is the number of FDA-approved drugs at 45% of them. And a lot of these patients also had many different mutations, exon 9, 11, as well as PDGFR and KIT mutations. So again, this is the response rate, and you can see it's pretty high at 26% across all patients. And what they saw, interestingly, was that the earlier you introduced the drug, the more patients responded, the more patients had a really significant tumor shrinkage. You can see here, for those who had just 2 prior TKIs, all of them responded. Again, only 2 patients, so small sample size. If you had 3 prior TKIs, it was 70%, but if you had more than 4, it was only about 6.7%. So I think it also shows that the longer you're on treatment or getting more in different inhibitors, the more resistance mutations you develop, and the harder it is for some drugs to work. Here's the waterfall plot, again showing patients on an individual basis how well they responded across many different dose levels, which is the color coding. You can see that about 26% of patients had a major shrinkage, more than 30% of tumors shrank. This happened across many different mutations, which you can see on the color code here at the bottom. And in terms of side effects, this actually had a little bit of a different side effect profile. Interestingly, this drug actually had a lot more decrease in blood counts, which you tend to not see as much on imatinib or sunitinib or regorafenib. You can see here that more patients had anemia, neutrophil count decrease, white blood cell count decrease, and in some cases these were pretty extensive. So I think this will go along to trying to find the right dose, because maybe the 40 milligram, which is the highest dose they test, was a little bit too much. Interestingly, they also had a lot of increase in the muscle enzymes, which sometimes can be seen with muscle damage. So this trial is open to accrual. We're actually open at Stanford as well for this drug. So we're happy to see any GIST patients, and again, this is accruing across many different lines of therapy. So conclusions are, there are exciting new drugs. These two new drugs are really exciting. They have broader KIT resistance mutation activity, and they're here now, and they're in clinical trials. What will be the interesting question is how these new drugs fit into the landscape of current GIST paradigm. Will they be second line? Will they be third line? Will they be fourth line? Where will it fit in? And I think that's where the clinical trials will really try to establish what the best place is for these new drugs. All right, thank you very much.